Welcome, everyone. My name is Sri Natarajan. I'm a professor here at UCSF in the Department of Radiology. I'm also a faculty member in the Joint Graduate Program in Bioengineering between UCSF and UC Berkeley, and a member of the Center for Intelligent Imaging and the Baker Computational Health Science Institute. Today, I'm here to talk about myself and my research program and that I do at UCSF that I'm involved in. So this short talk, the first part will be an introduction to my research interest, and then I will give you two examples of recent research studies coming out of my laboratory. My research interests broadly fall under two categories. I'm trained as a bioengineer, and in my bioengineering research, I'm mainly involved in developing machine learning algorithms for different types of structural and functional brain imaging. I'll give you some examples of that in the next slide. But basically, I've written a couple of textbooks about one form of brain imaging called electromagnetic brain imaging, and how we can use machine learning algorithms to improve this form of brain imaging. And more recently, I've been involved in machine learning algorithms for combining different types of brain imaging modalities. My other research interest is in neuroscience. As a neuroscientist, I do two kinds of neuroscience research. One is basic neuroscience research. I'm trying to understand, I'm very interested in looking at plasticity in brain networks, how brain networks change over time, but specifically brain networks that are involved in speech, language, and different types of cognitive processing. The other big part of my basic neuroscience research interest is trying to understand how the brain controls the way we speak. Speech is a facile behavior that is uniquely human, and how do we control the way we speak is something that's of great interest to me. This basic neuroscience work that I do is highly translational, or it can be basically translated into clinical applications. So my clinical translational work is really combining these sort of bioengineering tools for analysis of brain imaging data with the neuroscience tools applied to a variety of neurological and psychiatric disorders, ranging from developmental disorders like autism to disorders in the elderly, which are basically dementia. Today, I'm going to mostly focus examples of research that I've been involved in the area of dementia. So what we do in the lab is basically different types of or modalities of brain imaging. And one of the modalities that we use is called magnetoencephalographic imaging, which is basically recording magnetic fields generated by the human brain. We also do some invasive work like electrocardiography, where we have collaborators who are neurosurgeons who can place electrodes on the brain, and we can do direct brain recordings. We are also involved in functional magnetic resonance imaging and PET imaging with collaborators. We also do other kinds of imaging, which are basically manipulating brain excitability non-invasively. This is one example of this is called transcranial magnetic stimulation. We're also involved in different types of MRI imaging, as well as diffusion MRI or diffusion imaging, which is used to reconstruct the wiring diagram of the brain. Students and fellows in the lab have, over the years, been very interested in applications of brain-computer interfaces, looking at computer games and how they may change brain function and behavior in both in health and in disease. So I'm going to give you an example of my bioengineering application. One advantage of using machine learning algorithms in brain imaging is you can really rapidly speed up data collection. What I'm showing you here is our ability to reconstruct part of the brain that responds to touch on the skin surface. So this part of the brain is called somatosensory cortex, usually to extract reliable information about where in the brain is somatosensory cortex and when does somatosensory cortex get activated in response to a stimulus, you need to repeat a stimulus many, many times, typically about 250 times. You tap someone's skin and you average the brain response and figure out where it's coming from. That's what we typically do clinically. What machine learning algorithms can do is to really speed up this sort of data collection by looking at reconstructions of, say, a single tap shown on the right side here, or say 10 taps. It's basically giving you a 20 times speed up of the time duration of the experiment. And you can see here, these algorithms are able to actually accurately still reconstruct the location as well as the timing of somatosensory cortex, with even though the data was collected in less than 20 to 200 times less duration. So this is great advantage because we can now start to do experiments in little children or elderly who might be very fidgety, who may not want to hold still in the scanner for a long time. This is one of the advantages of using machine learning in brain imaging. I'm going to give you two other examples of brain activity and brain function that we can measure. Both are in the area of dementia of the Alzheimer's disease subtype. The first experiment I'm going to show you is looking at resting state brain oscillations in Alzheimer's disease patients. So when you are resting, just awake and not responding to any stimulus, the brain is actually active and we can reconstruct the brain activity. Not only is it active, their brain networks are synchronized, different brain regions synchronize their activity across very large, distant parts of cortex. And this synchrony is something we can measure with imaging, and we can reconstruct this synchrony. And what we show is that there's a frequency-specific abnormality in neural synchrony in Alzheimer's disease. This figure shows that these brain regions in the back of the brain, what are called as posterior brain regions, when you look at 8 to 12 hertz, which is also referred to as the alpha band, they actually show reduced synchrony. The color scale here is blue, shows reduced synchrony compared to a cohort of age-matched controls, and the red to yellow scale shows increased synchrony compared to age-matched control. What you can see is that if you look at 8 to 12 hertz, there is reduced synchrony. But if you look at 2 to 8 hertz, a band called delta or theta band, you actually see increased synchrony. This kind of frequency-specific modulation or changes in synchrony is information that is very similar to what has been seen in radionuclide PET imaging. But this particular data is obtained by having subjects come into our lab and just sit in front of a magnetic field scanner and measure their magnetic fields and reconstruction of this brain activity. So it does not involve any radionuclide imaging. So we think that this could be very exciting as an application to measure the processes that are abnormal in patients with dementia. One question that might ask is this related to disease severity? And so this is the data that we would normally obtain with PET imaging. And what I just showed you is that this kind of information can now be obtained with functional imaging using magnetoencephalographic or magnetic field-based imaging. And what I'm showing you here is that this reduction in alpha band synchrony is actually more severe. There are greater reductions in patients who have greater deficits. What is shown here is the MMSE score is on the x-axis is an assay of disease severity. So if you have very low scores, you have much more severe impact of the disease. And what we show is that patients who have much lower score, more severe, are the ones with greater reductions in the alpha band. But the same kind of disease severity effects are not seen in the 2 to 8 hertz range. This again suggests that there's a frequency-specific functional consequence of neural synchrony that we can measure. And we're now looking at different subtypes of dementia. Even within Alzheimer's disease, there are many subtypes of Alzheimer's disease. We're looking at both subtypes of Alzheimer's disease and subtypes of other forms of dementia. As I said, in our research interests are looking at neural oscillation during speech and language production. So we can look at these kinds of brain oscillation during speech and language production. And I'm going to show you an example of an experiment we do in trying to understand how the brain controls the way we speak. One way we can do it is to alter the feedback that subjects hear when they speak. So here's a subject in one of our scanners. Subject is supine, which is they're lying on their back, and we ask them to speak, and a microphone picks up their speech, and a digital signal processor can alter their speech in real time so that what they hear is not exactly what they produced, but some altered version of what they produced that we can manipulate experimentally. One manipulation we can do is manipulation of their pitch. Here's the same subject. They're just we just flip them so they're looking like they're sitting up. They're speaking. The microphone signal is shown in red. It's passed on to a digital signal processor, and the blue trace tells you what they're listening to, what they hear. And what we can basically do is experimentally alter, and from what they produce, we can alter it so what they hear is different from what they produce, and see what happens in the brain. One example of such alteration we can do is the alteration of pitch. So subjects would basically we ask them to produce a constant pitch. So they would go, ah, and we can lower or raise their pitch. In this particular experiment, we lower their pitch. So they produce, we tell them to produce a constant pitch. And when they're producing their pitch, what they hear is a lowering of pitch. So what they will hear is ah, and we lower it by one key in a piano. So if you have two adjacent keys, we lower it by one of those keys. And what, unbeknownst to them, when they hear a lowering of their pitch, and they didn't want to do it, they actually will raise their pitch to oppose this experimentally imposed lowering of pitch. This is their pitch control system in action. Now we do this when they're doing, when we're measuring their brain activity, and so we can reconstruct this activity. And anyway, we show that patients with Alzheimer's disease actually have an elevated compensation compared to controls in their pitch. And when we look inside their brain, shown here are controlled patients, and shown in the bottom are Alzheimer's disease patients. And we can measure activity dynamically in this period from 0 to 400 milliseconds. And what we can show is that patients with Alzheimer's disease do not activate certain regions like the left frontal part of the brain. And in compensation, they over-recruit these right posterior parts of the brain. And that's what contributes to their abnormalities in this particular pitch control behavior. So I've shown you a lot of information. Just to summarize, my research program is on imaging of brain oscillations and linking brain structure, genes, and behavior. I've shown you abnormal resting state neural synchrony in Alzheimer's disease patients that are related to disease severity, amongst other things. I've also shown abnormal neural oscillations during speech motor control that we can measure in Alzheimer's disease. Everything I've said is really representative of incredible team science efforts that goes on at UCSF. And I'm thankful for an incredible team of scientists who I've had the privilege and honor of working with over the years. And I look forward to continuing doing so. Thank you very much.